So hello, and thank you for joining today. I'm going to be talking today alongside my colleagues Rui and Matt to talk to you about real-time home-based analysis of microbiome metabolites using the OMED Health Breath Analyzer. And we're all very excited to talk to you about this today. So to give you a bit of context on who's talking today, there's myself. I have a technical background in biochemistry and biophysics, but I've spent the last 15 years applying my passion for exciting science to help more in the commercial space. And specifically, that's helping scientists to understand and benefit from very novel technologies. I'm currently heading up our business development activities in Europe and Asia for our research services business. And I'm honestly particularly excited about the OMED device and the benefits it could bring to the scientists I'm speaking to every day. It's got some really nice, unique advantages. We've also got Rui Lopez, who's a clinical medical doctor by background. So Rui's clinical experience as a physician has been used to help him develop a career in both clinical development and research. And he's worked extensively in gastroenterology, respiratory disease, and several other therapeutic areas as well. Rui is now leveraging his clinical expertise as a medical advisor for Owlstone to guide and advise the development and implementation of healthcare products within our company. And then finally, we've got Matt Kerr, who's also got a scientific background. His is in molecular and cellular biochemistry, and he's leveraged this across metabolic disease, cardiovascular disease, and neurological disease so far within biopharma and biotech. Matt's now sitting within the clinical biomarker services team at Owlstone, where he's primarily responsible for the analysis and biological interpretation of putative biomarkers across our clinical studies we're running. So before we get into the meat of the slides, I'm going to give you a quick overview about what we're going to talk about today. First, I'm going to give you a high-level introduction to Owlstone in terms of what we're doing as a company. I'm then going to give you a bit of an overview about what the OMED device actually does and what it's measuring. As a clue, it's hydrogen and methane. We're then going to hand over to Matt. Matt's going to give you a bit of context into the biology around hydrogen methane, which are the molecules we're looking at in the OMED device, and how they link into metabolism. And then Rui's going to take over and help us to understand a little bit about how hydrogen and methane are actually linked to health and disease. And he's specifically going to focus on gastrointestinal disease here. Rui's also going to talk a little bit about how people are currently analysing hydrogen and methane in breath, which leads me and then to talk about how the OMED device can be used in clinical research and how it's a little bit different than what's previously gone before. And I'll specifically focus here on some of the unique benefits that we think can be enabled using the OMED device in clinical research. And then finally, we're going to hand back over to Matt, who's going to give us a bit of interesting thoughts into where else hydrogen and methane, outside of just pure gastrointestinal disease, where else those molecules might actually be interesting to study, to give you some interesting ideas about where else could this kind of technology be applied in research. So first, a quick high-level introduction to Owlstone. So first, the kinds of molecules we're looking at in breath, we're looking at the gaseous phase molecules. So those are volatile organic compounds, things like acetone, things like short-chain fatty acids from microbiome metabolism, etc. But we're also looking at gases like methane and hydrogen. And those are the really molecules we'll focus on for the rest of the day today. So Owlstone as a company, we are about 200 people based primarily in Cambridge, UK, although we do have a few colleagues over in the US as well. And we are active in broadly trying to get breath and breath tests to clinical use. So we work in two different ways. Firstly, we work on the discovery side of things in terms of finding novel biomarkers, helping other people to find novel biomarkers that could be used to ultimately turn into a clinical test. And we've got lots of technologies to help and support in the discovery side of things. So, for example, some very reproducible ways to collect breath samples and this novel breath atlas we're developing, which helps people to understand what are the types of molecules in breath and what are their ranges. Once we've identified specific molecules that are particularly interesting to look at, we can then go on to the deploy stage. And Owlstone has several projects in development that could ultimately turn into clinical tests in, for example, cancer and liver disease. And of course, the other aspect of deployment we'll talk about mostly today is this OMED device, which we've just launched for use primarily in gastrointestinal health, but you'll hear today it could be used in some other areas as well. So what is the OMED device? So the OMED device is this little handheld thing here that you can see on this screen. It works in concert with an app. And this device basically quantitatively analyzes hydrogen and methane in the home environment. It's a device that can be used per person. So it's a thing that you send to an individual, and they can take multiple measurements of hydrogen and methane per day. Really, your only limitation is there has to be a 15-minute interval between different breath samples. There are no consumables needed. So it's really a one-off cost for the actual device. And then you can take as many samples. There's no additional charge for any additional analysis there. The app that it works in concert with, it allows you to first, of course, log the hydrogen methane, but it also allows you to log other things that we know can affect hydrogen methane levels. So the food and drink you're eating, the exercise you've done, the sleep quality you've had, and various other aspects of your lifestyle, as well as, of course, being able to track symptoms. So things like how bloated you are, what stool looks like, etc. In terms of the data we're collecting, as you'd expect for a device like this, it is absolutely secure and reliable in terms of the way that the data is stored. We're fully compliant with the regulatory authorities in the UK, the EU, and the US in terms of kind of storage of that data. And then the other thing that I want to just highlight here is the other thing we're developing is an app and a portal support specifically in research use. And this is very much in development at the moment. This app that's specifically for research use is going to do things like hide the hydrogen and methane levels to patients, because we know that there's a gut-brain axis going on as well. What we're envisioning this research use app and portal should allow us to do is things like compliance monitoring in trials. So you'll be able to see in real time whether patients are actually taking samples or not. It should also allow you to do a detailed interrogation of the data you're collecting at the end of the project as well. So I want to give you a quick overview of what it actually takes to give a breath sample. It takes under 3 minutes in total to give a breath sample. The first thing you need to do is actually connect the device to the app. There's then a 2-minute warming up period for the sensors to get to temperature within the device. Following the warm-up period, the patient would be directed to deeply breathe and then hold their breath for 15 seconds. And then again, at the direction of the app, they'd exhale gently into the device for 20 seconds. The idea with that is we really want to be analysing the breath that's been in most recent contact with the bloodstream. That's generally in the lower airways. So by collecting a breath sample for 20 seconds, you can analyse the sample towards the end of that breath, allowing you to get a concentration of hydrogen and methane that's from the bloodstream. At the end of that, you then just click view and save on the breath sample itself, and then that data is then stored into the cloud data storage. So that's a little bit about the OMED device. We'll talk a little bit more in detail about that later, but I'm going to hand over to Matt now to talk a little bit about hydrogen and methane and where they fit into biology.
Brilliant. Thank you, Liz. Hi, everyone. I'm Matt, and I'm a biomarker scientist here at Alstone. And I'm going to spend the next couple of slides talking firstly about where hydrogen and methane are coming from in the body, and secondly, why we're particularly interested in these two gases. When we talk about gas production in the human gut, we're talking on the order of magnitude of 0.2 to 1.5 litres of gas per day. And about 99% of that gas is either hydrogen, methane, or carbon dioxide. Carbon dioxide is systemically produced across the body, so our focus here is on hydrogen and methane. Starting off with hydrogen, the primary gas produced by the gut microbiome. In fact, if we look at gut microbes, we can see that around 70% are able to produce hydrogen. And in particular, Bacteroidetes and Firmicutes are the responsible phyla for hydrogen production. When we tack over to methane, we see a slightly different story. Rather than bacterial production, we have archaeal production, in particular, Methanobrevibacter smithii, making up around 95% of methane production in the average high methane producer. In fact, if we look at the gut microbiome for a high versus low methane producer, we see around 1,000-fold elevation in M. smithii in the high versus low producer. So we start to understand what is responsible for this gas production, but how are these bacteria and archaea producing hydrogen and methane? Well, as is the case for most gut fermentation, our starting input is indigestible fibers or polysaccharides. The primary mechanism for hydrogen production is our standard glycolytic pathway, generating reduced NAD and FAD being oxidized to produce hydrogen gas. And we see downstream from this pathway some of the other metabolites we associate with gut fermentation, such as short-chain fatty acids, propionate, acetate, and butyrate. We see a lot of hydrogen production on our way towards these short-chain fatty acids, which leads to these high hydrogen levels in the gut. And these can be further metabolized, particularly by methanogens, converting hydrogen and CO2 into methane in the gut as well. This has two important implications for the gut. Firstly, reducing the total level of gas, going from 5 moles of hydrogen and CO2 into 1 mole of methane, but also reducing the total levels of hydrogen, therefore reducing some of the end product inhibition of glycolysis we can see from the rest of the process. And with that, I'll hand over to Rui to walk us through some of the microbiome in health and disease.
Great, thank you, Matt. Hello everyone, I'm Rui and I'm a medical advisor here at Alstone. Matt has discussed so far the particulars of hydrogen and methane and its sources in the human body. And in the next few slides, we'll be exploring the critical role of the gut microbiome in human health, the importance of maintaining homeostasis, and the implications of dysbiosis in gastrointestinal diseases.
We know that the gut microbiome consists of trillions of microorganisms that play essential roles in digestion, absorption, immune function, and even mental health.
A diverse and balanced microbiome is associated with better health outcomes, while disruptions in this balance can lead to a variety of health issues. The microbiome is not an inert onlooker, but in turn it interacts with the host in a variety of complex ways. Eubiosis is this state of harmony and balance in the gut microbiome where beneficial bacteria predominate and support overall health. And in contrast, dysbiosis reflects an imbalance in the microbiome composition, with potentially harmful bacteria outnumbering the beneficial species, leading to a negative health consequence. There are many factors as well that come into play, such as diet, lifestyle and medications, and even environmental exposures that can disrupt the balance of the microbiome, tipping the scale towards dysbiosis. And it's this dichotomy of eubiosis versus dysbiosis that we will focus on to understand how hydrogen and methane behave when key changes happen in the structure and composition of the gut. The levels of hydrogen and methane produced by the gut microbiome and excreted in breath vary on a day-to-day basis and are influenced by several factors in a healthy population. Understanding these dynamics provide insights into the role of the gut microbiome in human health and in metabolism. So we know that hydrogen is produced when unabsorbed carbohydrates reach the colon and are fermented by commensals of the large intestine, while methane is produced as an escaped metabolism of hydrogen by archaea, and not every adult is a producer. So what do we expect in a normal population? At baseline, in fasted conditions, research shows us that hydrogen has a median value of 6 ppm and methane of 4 ppm. After meals, especially those rich in carbohydrates, there's a noticeable increase as a postprandial peak in hydrogen and methane production due to fermentation. Research also suggests that hydrogen and methane levels follow a circadian rhythm and are influenced not just by dietary composition, but by other factors. Variations in the type and abundance of gut bacteria among individuals can affect hydrogen and methane production. The speed at which the food moves through the digestive tract can also influence microbial fermentation. And finally, psychological factors such as stress and anxiety can have a tremendous impact on gut motility and microbial activity. So what do we observe when the scale tips towards dysbiosis? In a variety of gastrointestinal diseases, the levels of hydrogen and methane that are excreted in breath can significantly differ from normal values. For example, hydrogen may be produced in larger amounts in malabsorption syndromes, and if there's an excess of bacteria in the small intestine, such as in SIBO. And what we observe are specific patterns during hydrogen breath tests. For example, a rapid rise and an earlier than expected peak when the small bowel is colonized by bacteria. For methane producers, they may not exhale as much hydrogen as it's being converted into methane, and they traditionally have higher baselines and an earlier methane rise on breath tests. In GI disease, these levels of hydrogen and methane can therefore serve as biomarkers and indicators of the underlying conditions and link with the symptoms presented by the patient. So for example, conditions such as lactose intolerance or malabsorption syndromes can lead to altered fermentation patterns and changes in hydrogen and methane levels, which, when there's clinical suspicion, we can diagnose via hydrogen and methane tests. In IBS, which is a disease characterized by abdominal pain, bloating, and changes in bowel habits, hydrogen and methane levels may be altered. High levels of methane have been linked to decreased intestinal motility and are associated with constipation, especially in patients with IBS type C. Hydrogen accumulation due to bacterial fermentation of carbohydrates in the small intestine also contributes to bloating and abdominal pain, which is experienced by patients with SIBO. And I would like to make a particular mention of SIBO as a condition that has been extensively studied in the context of the microbiome and gas production, and where hydrogen methane and its utility as biomarkers is well characterized. So SIBO, which stands for small intestinal bacterial overgrowth, is a condition in which the small bowel is colonized by an excessive amount of aerobic and anaerobic organisms that are usually present in the colon. And it's this overabundance of organisms that results and causes gastrointestinal symptoms such as bloating, abdominal pain, constipation, and diarrhea, and in extreme cases can cause intestinal inflammation and malabsorption. The exact prevalence of SIBO is unclear. However, SIBO has been linked to several gastrointestinal and extraintestinal diseases. SIBO can occur in association with anatomical abnormalities, such as diverticular, fistulas, post-surgery in a surgical blind loop, in an ileocecal resection, been linked to motility disorders such as scleroderma, IBS, functional dyspepsia. It's been linked to metabolic and systemic disorders such as diabetes, disorders of gastric acid production in the stomach, chronic pancreatitis, cirrhosis, and even immune disorders. And a very popular and accessible tool for understanding how these gastrointestinal conditions, including SIBO, behave is through the representation of hydrogen and methane in hydrogen-methane breath tests, HMBT challenge tests. They are simple and non-invasive and safe ways to start to unravel the biology behind an individual's GI symptoms. And they're commonly performed in people who have IBS to rule out other underlying conditions such as SIBO or carbohydrate malabsorption. They've been extensively used in clinical practice and in research over the last 30 odd years, and its methodology is very well characterized. These HMBT tests provide a very practical way of studying a person's microbiome in a very standardized way. They reduce external factors to a minimum through general requirements such as controlling the diet the night before, fasting, restriction on the use of antibiotics, probiotics, restrictions on exercise, among other restrictions. And they allow collection of breath samples at multiple time points in a very standardized way to explore how hydrogen and methane change in response to a substrate challenge. Now I'll hand over back to Liz, who will further discuss on the capabilities of the OMED device. Thank you.
Thanks, Rui. So HMBT challenge tests, like Rui's just been talking about, are used regularly for the diagnosis of conditions like SIBO and carbohydrate malabsorption. And they present a really great way of diagnosing these conditions using just a few hours of breath testing on a single day, enabled really by the standardization of those external factors. And to do the analysis of hydrogen methane, they typically use highly accurate and sensitive analytical techniques in a clinical lab to analyze each breath sample that's being collected. However, we know from research that without controlling strictly for these external factors, there is significant variability in hydrogen and methane production and excretion, not only between different people, but also within the same person across different days. And this isn't surprising, as we know these gases have been consistently shown to be influenced by diet, particularly by carbohydrate ingestion, and also by emotional factors such as anxiety and also physical activity. And this high variability presents challenges for research specifically, where you might want to understand hydrogen and methane challenges in response to, say, a particular foods in a more real world setting, so without strict standardization of diets and lifestyle. Or, for example, when you want to understand the impact of an intervention like a probiotic on hydrogen and methane production. So to better understand the real world conditions and the complex interactions of hydrogen and methane with these external factors, we really need to collect repeated measurements of hydrogen and methane, not just over a day, but over multiple days. And the analytical techniques that are currently used in HMBT challenge tests would be suitable for this. They're accurate and sensitive enough, but the costs of doing repeat measurements like this would make longitudinal analysis extremely expensive. So what the OMED device makes it possible to do is essentially, because you don't have any extra cost per sample you're analysing, it makes it possible to build a really, really rich, detailed understanding of the hydrogen and methane dynamics, both within a single day, but also over multiple days. And to give you an indication of what that looks like and what that can look like over time, I've just plotted some of my own data here on the right-hand side. So on the top, you can see the average hydrogen level per day, and you can see, as you'd expect, there's quite a large variability against me when I'm eating my background diet. You can see this potentially even more on the lower slide, where I've actually plotted every single hydrogen reading I took over that 2-week period. And you can actually see here the kind of individual daily spikes of my hydrogen levels going up and down. And again, you can see some quite significant shifts in terms of some days I've got very high hydrogen, some much lower. So repeat sampling is really possible here. This ultimately helps you to average out some of those sources of biological variability, as Rui was talking about things like diet, circadian rhythm and exercise, which could help you ultimately to better understand the true impact of any intervention or challenge you might actually be giving in a research study. So I want to give you a few examples of how this could be used. Say, for example, you are wanting to understand how a fibre is fermented. Repeat challenge tests over multiple days so that you're essentially looking at a few different time points, averaging out some of that natural variability and actually getting a better picture of the average response to that particular fibre. That's particularly useful when you say want to compare between different fibres and compare between how they're fermented. You can get an average picture of how those different fibres are fermented and compare and contrast in a much more statistically significant way. And then the final thing that we think could be very useful about this OMED device is one of the ways that people traditionally do hydrogen and methane tests and use them in studies to kind of explore probiotic efficacy and things like that is they do a challenge test at the beginning and then a challenge test at the end and actually see, has the probiotic reduced the impact of that challenge test on the amount of hydrogen and methane being produced in response to a specific challenge. Now, as Rui's just talked about, if you're relying on a single point measurement, they can be very variable, which means you can get a lot of noise in there. And actually seeing a statistically significant change pre and post intervention can be quite challenging. What we're proposing potentially doing here is essentially avoiding a challenge test altogether. This also makes it more like the real world, which is, I think a lot of people are saying to me that they'd like to be able to look at it a little bit more. So, for example, with this kind of approach, you could take samples from patients over multiple days and really build a good understanding of what that person's natural variability, given their normal diet and their normal lifestyle, what their natural variability of hydrogen and methane looks like before you give them any intervention. And then post-intervention, you can again build up that understanding of post-intervention. What does their natural hydrogen and methane variance look like, given their natural diet and their natural environment? And to give you a nice example of what this can look like, this is an individual which we've seen recently where we've actually seen a response to potentially treatment. So this is showing the average daily hydrogen measurements, all hydrogen measurements on the bottom. So the average daily hydrogen, you can see quite nicely here. There's, again, a lot of variability in this measurement, as you'd expect, per day. The hydrogen methane spikes are quite different per day. But after this treatment have been sent, in this particular case, it was an antimicrobial treatment, you can see that actually the average hydrogen has dropped down substantially. This is potentially even more interesting when you look at the all hydrogen measures. So instead of averaging out the hydrogen per day, you're looking at all the hydrogen methane readings taken. You can see this kind of natural daily spike pattern happening still. And you can see a very pronounced shift in how that happens pre and post antimicrobial treatment. So pre, you've got very high hydrogen spikes coming basically every day. Post, much lower hydrogen spikes and not coming every day. So a very substantial difference that we can see here pre and post treatment to the hydrogen levels. So I hope that gives you a bit of context into kind of how this kind of technology might be useful in terms of understanding the impact of different fibres on fermentation and averaging out the natural variability, and also giving you some thoughts about how you could use this technology to actually look at efficacy of, for example, a probiotic or a prebiotic or a postbiotic or whatever your intervention treatment is, giving you a better way of being able to understand in a real world way what actually is the impact of your product, and can you see something? So the other big benefit that we can see of the device that we're using here is something I referred to at the beginning, and it's really decentralized testing, but decentralized testing with compliance monitoring. So with the fact that all this data is being collected and uploaded to the cloud instantaneously, you can essentially get a picture of actually what is being collected in your study as it's going. So this gives you on this slide an example of kind of what this data can look like. So this is again data taken from me. So I can see here all the individual samples I've taken. It shows when I've taken them, and then you can actually use the data interrogation software to allow you to see how many samples I've taken per day as well. So if you, for example, have a particular target of you want your patient to take 4 to 6 breath samples a day, you can look in this data and you can see quite quickly if someone is actually complying with that or not. And if not, then you can go and amend that with them, go and have a discussion with them. Supporting this decentralized trial use as well. Some of the other things that we're measuring in the app might also be quite useful for monitoring in clinical studies and might provide kind of a single point of use for the patient to interact with and make the clinical trial just that little bit simpler. So I did talk about this earlier in the presentation, but I wanted to talk to you a little bit more detail about how this works. In the OMED app, you can record food and drink details very quickly and easily. You can set the time and date as you need for when you actually ate the food and drink. You can record symptoms. So we have a 1 to 9 scale in bloating, in abdominal pain, in nausea, in flatulence. And then with stool, we use the Bristol stool scale in order to help the patients to classify what kind of stool they've had on a particular day. And again, you can set the particular date or time that those symptoms were observed in the app as well. I also talked about the other kind of things that can impact hydrogen and methane readouts. So that's stress, sleep and exercise. And again, we've got a sliding score here to help people record on a particular day and what their lifestyle looked like on that particular day. And all of that data is then available for analysis and viewing in the portal as the patient collects the data and the study goes on. So the final thing I'm just going to touch on is I talked about this breath atlas at the beginning. That's largely been so far for VOCs specifically, so where we're looking at the particular types of VOCs we see in breath and their levels. But of course, this atlas will include hydrogen and methane data as well. So one of the other things that will come out of projects when you're doing them with us, when you're doing research projects with us, is there'll be this huge data set and reporting on kind of the normal ranges and changes in hydrogen and methane that we can compare your data against and help to understand a little bit potentially more biological context into what might be going on in your particular study as well. So I hope that helps with giving you a bit of a broader picture in terms of how the OMED device is a little bit different than what's gone before and potentially gives you some ideas about how you could use it in your own research and clinical studies. I also just wanted to touch a little bit on how the device actually works and give you some confidence that when you use this device, you'll get data that's going to be comparable and usable in comparison to other ways of looking at hydrogen and methane as well. So first, to touch on how the OMED device actually works. As I said, this is a very small handheld device. Basically, what happens is breath passes through this device. It passes active carbon components, which filter out some of the interferent gases that might be present, might make it challenging to actually analyze the sample. So that cleaned up air then gets passed to 2 metal oxide sensors. And the passing of those gases past those metal oxide sensors essentially creates an electronic signal, which is dependent and proportional to each gas concentration. So in the device, those signals are then passed to an onboard processor where the gas concentrations are actually calculated by a machine learning algorithm that has previously been trained with literally hundreds of precise synthetic breaths to help it work out what the concentration of the hydrogen and methane levels are. And of course, then that information is then sent to the app. So to give you an indication of how this device stacks up against kind of conventional hydrogen and methane methodologies for analysis, this compares against the Gastrointestinal, which is a standard clinical use instrument to analyse hydrogen and methane in the clinic. So first, we took different readings from the Gastrointestinal itself and compared those readings against each other. So you can see here a perfect result would be essentially aligned to that green line in the centre. So you can see here there's a little bit of off-piece reflecting the hydrogen and methane levels when you compare those different samples together. When you're looking at the OMED device versus the mean of those two different samples, GG1 and GG2, you can see actually the data looks remarkably similar, which is already a very good sign. In terms of how that translates to accuracy and precision, you can see here, when we're looking at OMED versus HMBT, the OMED device is a little bit less accurate, a little bit less precise than I think the Gastrogenius, but it's really not very far off. And that's particularly impressive given that the cost of the OMED device is about 100-fold less than the Gastrogenius system. So it's not going to be quite as accurate or quite as precise as a kind of lab-based hydrogen methane breath tests, but it is going to give you the ability to collect very data-dense longitudinal information, helping you understand those hydrogen and methane dynamics. So a little bit of a different use case for this device, we think, versus the kind of clinical setup that a lot of people use. What I'm going to do now is just pass over to Matt. We've talked a little bit about OMED device and digestive health and given you some ideas there. There's potentially indications that hydrogen and methane might be useful in other places as well. So Matt's just going to talk to you a little bit about that and give you some potentially more ideas in terms of where hydrogen and methane could also be analysed in different research areas.
Brilliant. Thank you, Liz. So over the past couple of slides, we've heard a lot about the role of hydrogen and methane in the gut and gut health, as well as about the new OMED device, which allows for these longitudinal studies of breath hydrogen and methane levels. Over the next 10 to 15 minutes, I'm going to spend some time focusing more on basic science research, in particular focusing on summarising some of the exciting and emerging basic science research on methane, particularly on the production and potential roles of methane outside of those discussed already by my colleagues today. We'll address some of the challenges associated with this work as well, particularly how can we identify correlation versus causation in these settings, and highlight some areas where easy, novel access to epidemiological longitudinal data sets can provide additional insights into existing hypotheses. In particular, I'm going to be focusing on three separate areas. The first being closer to home and building on what Rui said today already about the link between methane and constipation and diarrhea. We're digging a bit deeper into the link between methane generation and gastrointestinal motility. Secondly, we'll be moving over to links between methane and metabolic status, particularly correlations between methane and BMI and cardiometabolic risk. And finally, we'll change tack completely, moving away from gut-produced methane and more over towards non-microbial potential endogenous sources for methane formation and the contexts in which that may be significant. So starting off with gastrointestinal mobility. And this won't shock anyone in the audience who's been listening to Rui speak earlier today. GI transit time is positively correlated with methane production in the literature, i.e. higher methane levels, slower GI transit. Importantly, this can be seen across the literature and across a variety of methods to measure this transit time, looking at either whole gut transit time or a cecal transit time, colonic transit time, or even stool frequency, with all of these papers showing an increase in GI transit time in methane producers versus controls, and the vast majority being significant or either close to significant for these values. So higher methane, slower GI transit, longer GI transit time. Again, teasing out correlation versus causation in this setting is challenging, but we can see papers where they've administered rifaximin to people with long GI transit times, noting that in 60 to 70% of cases, there were improvements in GI transit time after administration of rifaximin and removal of methanogens and those high methane levels. So potentially a causative link between methane production and long GI transit times in this setting. We can take this a bit further. Firstly, fantastic work by Pimentel et al., who removed any other effects from the gut microbiome. They administered methane directly via intestinal fistulae into dogs, demonstrating a 59% increase in GI transit time in the presence of methane versus in the presence of room air, measured by recovery rates of a radiolabeled tracer. So convincing evidence then that methane has a direct effect in elongating GI transit times. But what might be actually causing this? Well, Park et al. Provided the first potentially direct evidence for what might be driving this effect. They took guinea pig ileum under electrical field stimulation and measured the ileal muscle contraction, demonstrating across a wide range of frequencies almost a twofold increase in muscle contraction in the presence of methane versus control. This increased muscular contraction correlates then with increased GI transit times, providing this potentially causative link between the two phenomena. Moving on next to our second case study, correlations between methane production and BMI. There have been about 7 papers over the last 20 years or so which look at breath methane levels and BMI, some at baseline and some after a lactulose challenge. 5 out of those 7 papers found a significant correlation that higher BMI correlates with higher breath methane, such as this paper from Basari et al. Demonstrating an increase in 7 for your BMI for methane producers versus non-methane producers. Importantly, it's well understood that the gut microbiome is altered in obesity. So whether this is a direct link to methane production or indirect remains to be seen. Trying to understand why increased breath methane levels might correlate or potentially causatively correlate with increased BMI, we can start to look at what we've seen over the previous slides, namely that we know increased methane levels leads to longer GI transit times. These longer GI transit times give increased time for the host to uptake nutrients and calories from food in their diet. The second potential link between methane and BMI is short-chain fatty acids. And here we have to cast our mind back to one of the earlier slides where we talked about potential co-colonization of methane increasing potential production of short-chain fatty acids. This has been demonstrated in vitro with either just the hydrogen short-chain fatty acid producing bacteria on its own in yellow or co-colonized with methane producing bacteria in green, demonstrating significant increases in acetate levels in co-colonization versus single colonization. Interestingly, slight decreases in propionate as well. However, these increases in acetate in this in vitro setting correlate and transfer over to an in vivo setting, with mouse models demonstrating increases in serum acetate levels with co-colonization versus single colonization. Given that in an adult, around 10% of your calories in your diet come from short-chain fatty acids, it's reasonable to hypothesize that increased production and availability of short-chain fatty acids might lead to increased calorie availability for the host and increased BMI over time. So good indications that high methane correlates with high BMI. But this isn't necessarily always a negative thing. In fact, if we look at the gut microbiome in a pre-diabetic patient versus a type 2 diabetic patient, we can see that one of the most conserved changes from that pre-diabetes to type 2 diabetes is a decrease in bacterial methanogenesis in the gut, i.e. loss of methane seems correlated with that link between pre-diabetes to type 2 diabetes. And in fact, if we look at cardiometabolic risk factors, one of the big ones is visceral fat area. We can see that visceral fat was found to be lower in high methane producers, despite that increase in BMI. So methane is potentially protective in these high BMI settings. There's been a host of excitement in the last 5 years or so about GLP-1 agonists as a blockbuster drug in the treatment of type 2 diabetes. And interestingly, methane has been seen to increase GLP-1 secretion in both mouse glutag cells, here on the left, and human NCI H716 cells, with around a twofold increase compared to baseline in the presence of methane in both settings. And interestingly, this correlates to very preliminary data demonstrating increased GLP-1 in the blood of methane producers compared to non-methane producers after a meal in the morning. So methane potentially correlated with a high BMI, but potentially protective in that setting as well. With that, we'll come on to our third case study, and this is of non-microbial produced methane. Hypotheses for how this production could occur have been around since the early 2000s, with the primary one being standard metabolism in a mitochondria, generating reactive oxygen species as a product, i.e. hydrogen peroxide and hydroxyl radicals, and oxidatively demethylating methane-containing compounds such as methionine, resulting in methane production in vivo from oxidative stress. This hypothesis has been tested a number of times over the years by Geisy et al., providing some of the first in vitro evidence supporting this hypothesis. They firstly looked at the effect of in vitro hydroxyl radicals on some of those methane-containing compounds, demonstrating a significant release of methane in the presence of oxidative stress from some of these endogenous compounds. They took it a step further, taking this from in vitro to ex vivo, isolating liver mitochondria, putting them into a high oxidative stress environment and demonstrating significant release of methane over time with a linear correlation to the amount of mitochondria present. Finally, they're taking this step even further, demonstrating that endogenous ROS can do this as well, via application of sodium azide and cyanide, increasing ROS in these primary aortic endothelial cells and resulting in emission of methane in a dose-dependent fashion. So we've got good evidence that a hypothesis exists for methane production endogenously, and that this can be demonstrated in vitro as well. But these findings also translate to in vivo settings, firstly demonstrated by work by Kepler et al. In 2006, who took plant matter, particularly ash and beech, sterilized it to remove any microbial effects, demonstrating that all methane production was endogenous in nature, and then demonstrating methane production at baseline from this plant material. Translating them from plants into animals by Tuboli et al., we see work administering that same mitochondrial inhibitor, sodium azide, to whole animals and measuring whole body methane production. Walking through, we can see in our sham operated animals, in teal, no significant change in methane over the 8 days. When we administer this mitochondrial inhibitor, sodium azide, a significant increase in blue over the 8 days, and importantly, correcting for potential production of gut microbial methane by administration of antibiotics, we still see this elevation over the 8 days after administration of sodium azide, indicating this link is not because of alterations in the gut microbiome, but is because of endogenous methane production. These are obviously challenging experiments to do in humans, but Kepler et al. In 2023 took blood from human volunteers, incubated them with different concentrations of radiolabeled methionine, and showed that over the course of a day, you could see methane production from this radiolabeled methionine, i.e. this process can happen endogenously in humans as well. So we demonstrated a hypothesis in vitro, ex vivo, in vivo, and finally human settings, whereby oxidative stress can lead to methane production. But what may be the relevance of this in standard human physiology? Well, the first indications for this came in 2013 by Tuboli et al. They administered LPS to a mouse model, which is a mimic of sepsis or intense infection. And they demonstrated around a 2 to 3-fold increase in methane concentrations exuded from these mice after LPS treatment, i.e. that the high inflammation, high oxidative stress setting of infection had similar effects in increasing the production of methane by these animals. Studies are to date lacking in terms of large patient data cohorts to understand if this effect can be seen in humans as well. But fantastic data from Polag et al. In 2022 in the height of the COVID pandemic demonstrated that breath methane saw similar 2 to 3-fold elevations in response to testing positive for COVID-19. As the patient recovered from COVID-19 over the course of 8 days, we see his breath methane levels drop back down to baseline, indicating potential for breath methane to be a proxy, an indicator for infection in humans. So hopefully over the previous couple of slides, you've seen some exciting emerging research onto what effects methane might have in physiology outside of the gut as well. And with that, I'll pass back to my colleague Liz for a summary of today's presentation.
So I hope that's given everyone some useful context into how the OMED device can be used to support research in a variety of different ways. I wanted to summarise now, just to close off with kind of, telling you a little bit about the OMED device and what we've talked about, but also covering a little bit more about what Alstone do as well. So as a summary, Alstone are here really to provide tools to use breath in research and ultimately to help support translation to clinical use. We work on both the discovery and characterization side and the deployment side. On the discovery and characterize side, you can of course use this new OMED device for characterization of hydrogen and methane to explore it as a potential biomarker for novel applications and use it to understand efficacy for probiotics, prebiotics, and how fibers are fermented and things like that. We also, of course, offer technologies to do volatile metabolite analysis as well. So that can be things like short-chain fatty acids and other microbiome metabolites. That's broadly using our Omni service and is supported by that VOC atlas where we're gathering data, understanding what VOCs we see in breath and what levels they're at. And to give you a bit of context, these are some of the microbiome metabolites that we can see in breath that we measure in that standard VOC analysis platform. So if you would be interested in analysing broader microbiome metabolites outside of hydrogen and methane, that's definitely something we can support with too. And do just reach out and have a conversation with us. On the develop and deploy side, of course, we are quite active in that discovery space, trying to help people to use breath in research and identify and characterize new biomarkers. Once those new biomarkers have been identified, we also very much actively help people translate that to actually clinically useful tests. That's evidenced in that OMED device, which has actually been developed for clinical use to manage SIBO and generally in gastrointestinal conditions. We also have a test for liver cirrhosis. It's very actively going through development and clinical validation at the moment. Alongside our own work developing tests, so we do also work actively in partnerships with people. So just 2 examples I can talk about right here. We're working in partnership with the Defence Innovation Unit, for example, to help them develop ways to pick up pre-symptomatic infections. We're also working with the Bill and Melinda Gates Foundation on development of breath-based diagnostics for infectious diseases like HIV and TB. So if kind of developing clinical tests is actually what you're interested in, again, that's something we can support with, and please do just reach out to have a conversation and explore that more. So thanks very much. I hope you've enjoyed that, and we'll take questions now.
So I'll kick us off. So there's one question here. Maybe this one is for Liz. So I noticed that OMED app provides information on bloating. Is there a specific biomarker you look for to report this? Or is this self-reported by the patient? So maybe Liz, you could take that one, please.
Sure, yeah. So the way that the bloating logging works is it's a sliding scale based on 0 to 10. And the patient can basically select about where they are in terms of bloating. So, I mean, I've been using this device and using this app myself quite a lot as well to just experiment with it and check it out, etc. What I find is it's very easy for me to be my own control. So I know what my own bloating level feels like, and I've got an appropriate number that I put in when I feel approximately that level. So it is patient reported, but because it's kind of an individual tracking thing, you have a good degree of kind of correlation, at least within an individual patient.
Thanks, Liz. That actually leads into the next question, and maybe I can take it builds from that. So thanks for the presentation. From the data you've collected so far, how good is the correlation between the gases and reported symptoms, especially bloating, flatulence, and abdominal pain? So we don't have that data in the presentation, but we do record those symptom data routinely during the hydrogen-methane breath test. So that's the SIBO test that we've been running here in the UK for many years in partnership with Functional Gut Clinic. So it is important to look at those symptoms in conjunction with the breath gases. So we'll make sure that we include some of that data and share it with everyone when we send the slides out. But I think, Liz, you make an interesting point there that the symptoms are to a degree self-reported, and what one individual will rate at a particular level may be different from another. Next question that's come through, maybe this is for Rui. So if the device were being used without a substrate, can you provide some guidance on how many breath samples you might want to collect over a day if you are running over a baseline collect period, for instance?
Definitely. So what we've seen in literature is that there's a few papers that's been describing the circadian variation of hydrogen and methane between individuals and within the same individual. And one of them is Le Marchand et al., where individuals enrolled in the study have done a mean of about 14.5 tests per day across 2 days, which describes in detail this inter-patient variability. And he went on to suggest that to reduce the noise of discrete sampling, we should do a minimum of at least 4 samples per day across different days to be able to describe the baseline of those patients. And this ties up with depending on the requirements of the use for the device. So there's no limit. There's as many samples as you want to take. You can take, and the more samples, the better in terms of decreasing that noise. But it will always be dependent on protocols and requirements, and obviously patient compliance as well. but I would say that samples minimum is a recommended protocol in the literature.
Excellent. And there's one building on that. So it had been reported in literature before around single fasted methane measurements in the morning and how that can be beneficial. I was wondering if you could comment really on the utility of that initial measurement and how that may be helpful in things like IMO.
Yep, so definitely. So for IMO, we know that those patients that IMO producers have a higher baseline. But we also know that, and this has been described by a paper of Allegri et al., that depending on the time of the day they take the sample and the conditions, there's a very high variability of the values of both hydrogen and methane across the sampling. So having several samples as a fasting state will allow us to normalize that variability of methane, which is not as much as the variability of hydrogen, but it's still there. It will allow us to normalize that variability and then establish a confident diagnosis of IMO based on that mean level.
Excellent, thanks. Next question here, I think this one will be aimed at Matt. So I had only heard recently about this mechanism of endogenous methane production. It's fascinating. I think I'd only ever heard of it in the context of GI disease before. So the question here is, are there any potential physiological roles for methane in infection, or is it purely a product of oxidative damage?
Yeah, thank you. It's a really interesting question. And I think the short answer is the data is still emerging in this area. But to give a quick indication of where there may be relevance, so there's been some interesting indications from both in vitro and animal in vivo studies indicating that methane may have some anti-inflammatory, antioxidative, and anti-apoptotic effects as well. And in fact, if you translate that to some animal models of elevated oxidative stress, so here we're talking about sepsis models or ischemia reperfusion models, methane loaded saline solution in those settings can be seen to be cytoprotective, so potentially conferring some protective effects in high oxidative stress settings as well.
Great. There's another question that maybe direct this one to Matt as well. This is from Terence. There's reference to hydrogen-methane as a measure of SIBO bacterial activity. However, the proposed mechanisms by which hydrogen is produced relates to energy metabolism. You think the data collected by your device could be used as an indicator of energy metabolism or resting energy expenditure contributed by the microbiome (brackets anaerobic metabolism)?
Yeah, it's a really interesting question. I think I'll break that into two separate pieces, perhaps. First, talking about energy production by the gut microbiome. Secondly, how that might be related to the host metabolism as well. Over to the first part, so you're right that hydrogen and methane is a product of anaerobic fermentation and therefore talks about sort of microbial activity within the gut. I think the challenge of relating that directly to energy metabolism of the gut microbiome is separating out, firstly, the influx of substrate availability and correlations to hydrogen and methane in that setting, but also total gut microbial load as well. I think both those are going to have a significant impact on the levels of hydrogen and methane produced. So directly correlated to energy metabolism is perhaps challenging in that setting. The second aspect, I suppose, how does it relate to host metabolism? We touched on it briefly there. I think the major way it's going to relate to host metabolism is altering potentially levels of short-chain fatty acids and availability, therefore, for the host, rather than directly on host metabolism via glycolysis.
Thanks. There's one question, commercial question, maybe aimed more Liz. How is the OMED device compared to the R2 device? That's another product in the market at the moment. So maybe you could comment on that, please.
Yeah, sure. So I think the first thing to say is in Alstone, as I think you saw, I alluded to in the presentation, we spent quite a lot of time and effort really making sure that the device is as accurate and precise as it possibly can be. And we've tested literally hundreds of samples against our devices to make sure that they are as accurate and precise as we expect them to be. And we can share a post here kind of summarizing a bit of that process in the chat quite happily. There's not that much publication data out there on the Air 2, so it's kind of hard to judge how well the device compares to that. But what I think we can say is that if you look in the literature, if you look in the kind of people who are using that device, it does look like there's a little bit of inconsistency in the data it's providing. And again, we can share some links as to where we've seen some of that kind of reporting going on as well.
This next question, maybe this one is for Matt as well. So there's the different mechanisms of production of the methane. So how to separate arterial and somatic methane and exhaled? I don't think you can, is probably the answer.
Yeah, I think with those kind of questions, it's important to consider kind of the upstream of it. So if you're looking at a challenge test, obviously, therefore, you're mostly looking at microbial formed methane as a response to that. If you're in the context perhaps of infection, maybe expect more somatic. But I think it's important to consider the context around methane production as well as methane levels in that setting.
Thanks. Next two will group together. So there's a question about the 15-minute interval and also limit of detection. Maybe Liz, you could comment on those.
Thanks. I'll get someone else to cover the LAD, but for the 15-minute interval part, what I will say there is, that's primarily about making sure the device can be reset, and those kind of charcoal filters can be nicely cleaned up to make sure that the next breath sample isn't contaminated by anything beforehand. So breath is quite humid, for example. One of the things that that 15-minute interval allows us to do is just clean up that humidity. So that 15-minute interval is important. I mean, it's really the only limitation on the device, so in terms of how many samples you can collect.
Yeah, and the piece that I can answer is the relevant measurement range, as I say, is that low ppm to tens of ppm. That typically is where you will see the cutoffs as specified by the guidelines. So the training data that was used in the development of the device and then the validation work, that's typically the concentration ranges that you see. And that's quite unusual compared to other breath gases. We know a lot of other VOCs are typically at ppb levels, and it's quite difficult to detect at those types of concentrations using things like metal oxide sensors. So hydrogen and methane are a little bit special because they tend to be present in the ppm level on the breath. We've got time for one last question. I think this one's for you, Liz. How much is it?
I think what we'd quite like to do is have a chat to you about that one. So the research use piece especially, it is going to depend a little bit on the size of your study and things. So I think if you want to have a chat to us about that, please feel free to reach out to us. You can reach out to me at elizabeth.crone@alstone.co.uk, and I can direct that to the appropriate person if that's not me.
Excellent. Well, thank you. We're coming up on the hour, so just want to say a big thanks to the speakers and a big thanks to everyone who joined us today. So as I say, we'll make the slides and the recording available afterwards. There's a few links that we'll provide as well, so we'll be sure to do that. So thanks a lot, everyone, and have a great rest of your day, and hopefully see you again soon for the next webinar. Bye now.